Hello friends, I am Dr. Sanjula Babuta from Department of Pharmaceutics, School of Pharmaceutical Education and Research, Jamia Hamdard. Today, we shall be studying about the plasma protein binding. In this module, we shall be covering the various topics like the types of plasma proteins, factors affecting drug protein binding, methods of measuring plasma protein binding, the kinetics behind the protein binding and the significance of plasma protein binding. Now, let us understand what protein drug binding is. A drug when administered in the body can interact with the various blood and extravascular tissue components like proteins, DNA, melanin to form a drug macromolecular complex. Out of these components, protein interact with the majority of the drugs and this phenomena is known as protein drug binding. Binding of a drug can augment or diminish a drug's performance in the body. Lesser the binding to the various kinds of proteins, more efficiently a drug can transverse through the cell membranes. It is only the unbound fraction of the drug present in the blood which interacts with the various receptors, enzymes or can be transported through the various biological membranes. Even the metabolism of this unbound fraction would be extensive and would be excreted much faster as compared to the bound fraction. Therefore, it is very important to evaluate the extent of protein binding of drugs. The distribution of the drug in the body is regulated by various physicochemical properties of a drug, the binding affinity to plasma proteins and binding affinity to tissue proteins. A drug reversibly binds to the plasma protein and act as a reservoir for the unbound fraction which is readily eliminated from the body after showing the desired pharmacological action, thus prolonging the duration of the action of various drugs. The drug binding to the plasma protein is through the hydrogen bonds, ionic bonds, hydrophobic bonds or the van der Waals forces which are considered to be the weak chemical bonds and therefore, the binding is considered to be a reversible process. However, irreversible drug binding could also arise due to covalent binding resulting in the increased chances of carcinogenicity or tissue toxicity of the drug. Like for example, chloroform and paracetamol metabolites when bind covalently to liver could result in hepatotoxicity. Plasma protein binding greatly influences the pharmacokinetic and the pharmacodynamic parameters of a drug deriving from the fact that the bound drug is both pharmacokinetically as well as pharmacodynamically inert. Therefore, drug bound to the protein can neither be excreted nor be metabolized and does not show any pharmacological activity. Protein binding is considered significant for certain classes of drugs such as sulfonamides, anticoagulants, non-steroidal anti-inflammatory drugs, several compounds used in cancer chemotherapy and certain endogenous hormones like thyroxine and corticosteroids. There are a number of binding sites present on a protein. Competition for plasma protein binding sites between various drugs could lead to serious adverse reactions. For example, the interaction of warfarin and several sulfonamides with phenylbutazone. There are chances of inter-individual differences in plasma protein binding of many drugs due to genetic, environmental, pathological and other factors like aging. Types of plasma proteins. Various endogenous and exogenous molecules throughout the blood stream are carried by the various proteins that are present in human plasma. There are over 60 different types of proteins in human plasma, out of which albumin is the most abundant. Reactive groups originating from the tertiary structure of the proteins result in the binding of the drugs and other endogenous compounds. The law of mass action which proposes that the proportion of bound drug remains constant provided the binding sites are not saturated, governs the interaction between the protein and the drugs. Saturation of majority of the drugs within the therapeutic range is generally not observed, but the statement does not hold true as in the case of drugs like valproic acid, which indicates saturable binding at therapeutic plasma concentrations. The vital plasma proteins include albumin, lipoproteins and various globulins. The drug binds to the plasma protein in the following order: albumin followed by alpha acid glycoprotein, then lipoproteins and then globulins. Now, let us study in detail about the characteristics of these proteins. The first is human serum albumin, also known as HSA. This is a highly soluble and the most abundant plasma protein with a concentration ranging from 35 to 50 milligram per ml. It plays a vital role in maintaining the blood pH, the colloidal osmotic pressure and is accountable for most of the thiol content of the plasma. Human serum albumin has a molecular weight of about 65,000 daltons. It is composed of a single chain of 585 amino acids. It has the ability to bind to a large variety of all types of drugs and various endogenous compounds like fatty acids, bilirubin and tryptophan. It binds comprehensively to weak acids, neutral and weak basic compounds. Binding of small molecular weight molecules to human serum albumin guards itself from being eliminated through glomerular filtration of the kidneys and the various enzymatic reactions in the liver and the bloodstream. 4 different sites on human serum albumin have been identified for drug binding. The binding site for acid is generally the N-terminal amino acid, whereas the basic drugs bind non-specifically. Therefore, the capacity of albumin to bind to the acids is limited, whereas for the base it is quite high. The next protein is the alpha 1 acid glycoprotein, abbreviated as AAG, is the second most important plasma protein which generally binds to basic and neutral drugs. It is an acute phase protein having low molecular weight. Under normal pathological conditions, the concentration of alpha acid glycoprotein ranges from 0.5 to 1 milligram per ml of plasma. In certain disease states like inflammation, malignant disease, stress etc., this value can exceed up to 3 milligram per ml. The physiological function of this protein is not well known, but it has some immunosuppressive and immunomodulating properties. It also acts as an inhibitor of platelet aggregation, neutrophil activation and lymphocyte proliferation. There are a number of factors that affects the drug alpha acid glycoprotein binding, like the concentration of this protein in the blood plasma, heterogeneity of the glycan, AAG polymorphism, pH and competitive binding of AAG with other endogenous or drug ligands. It consists of a single wide and flexible site, and in some cases it is believed to have 2 overlapping sites. Here the binding is mainly by the hydrophobic bonds. It is an important binding site for basic drugs like imipramine, lidocaine, etc. The next protein is the lipoprotein. Lipoproteins having high lipid content bind to the lipophilic drugs through hydrophobic bonds. The concentration of lipoprotein in the blood plasma is variable and is much less as compared to the human serum albumin and the alpha acid glycoprotein. The drug binding to the lipoprotein generally depends upon the lipophilicity of the drug molecule as the drug has to dissolve in the lipid core of the protein. Different classes of lipoproteins are chylomicrons, very low density lipoproteins, low density lipoproteins and high density lipoproteins. Basic and the lipophilic drugs have generally higher affinity for lipoproteins. Various drugs like diclofenac, cyclosporin, chlorpromazine are example of certain drugs that show binding affinity for lipoproteins. The next plasma proteins are globulins, which have been identified in the blood plasma. There are different types of globulins like alpha globulin, it is also known as transcortin. It helps in binding many of the steroidal drugs, namely cortisone and prednisone. Therefore, it is also known as a corticosteroid binding globulin. The second is the alpha 2 globulin. It is also known as ceruloplasmin. It generally binds to vitamin A, D, E and K and also to cupric ions. Beta 1 globulin, it is also known as transferrin and it binds to ferrous ions. Beta 2 globulins, they bind to carotenoids, and the gamma globulins, which generally binds to antigens. The different types of plasma proteins and their characteristics have been summarized in table 1 as shown on the screen. Now, let us study the various factors that affect the protein drug binding. The first of all, we will study about the protein related factors. In the protein related factors, the major factor is the concentration of the protein in the blood plasma. This is the most important parameter affecting the protein drug binding. As human serum albumin constitutes the major portion of the human serum plasma, so most of the drugs bind to it. The number of binding sites which are available on the protein. In association with the concentration of various proteins which are available for the binding of various drugs, the number of binding sites over different proteins also play an important role in protein drug binding. The physicochemical nature of the protein. The various physicochemical properties of the drugs also play an important role in the drug binding process. Lipophilicity is the most significant parameter for the protein drug binding to occur in the body. With an increase in the lipid content of the drug molecule, the rate of protein binding process also increases. The second are the drug related factors, and under the drug related factors, the first are the physicochemical properties of the drug. Lipophilicity is the most significant parameter for the protein drug binding to occur in the body. With an increase in the lipid content of the drug molecule, the rate of protein binding process also increases. Then the second is the concentration of the drug. It also plays an important role in protein binding process. More the concentration, more is the binding if the drug has an affinity for a protein. The third is the drug protein or the tissue affinity. Various drugs, they show different affinity for different proteins in the body, which is considered to be a vital factor for the drug protein or the tissue binding. For example, digoxin has an affinity for cardiac muscle. The next are the drug interactions. Interactions occur when there is a competition between the drug and the other substances for a particular binding site over the protein. Alteration in the concentration of a protein by any substance leads to modification of the affinity for the drug molecule. The next are the pathophysiological conditions. Drug protein binding may be affected severely in disease conditions such as renal failure and hepatic failure. In both the cases, there is decreased albumin content, which results in decreased binding of several acidic drugs. Now, let us study the various methods which are used for the measurement of plasma protein binding. Many successful methods to measure the degree of plasma protein binding of drugs have been derived. All these methods work by physically separating the unbound drug from the bound drug and calculating the relative fractions. Some beneficial techniques to measure the plasma protein binding are equilibrium dialysis, ultrafiltration, ultracentrifugation, gel filtration and microspheres of albumin. There are some other techniques also which are utilized to a lesser extent, such as micro extraction and optical biosensors, which have been utilized to measure the highly protein bound drugs. None of the above methods could be judged as superior over others, as all these methods possess some advantage as well as some disadvantage. So, let us study in detail about them. The first is equilibrium dialysis. It is a well accepted and a popular method used in plasma protein binding. This method employs a semi-permeable membrane. On one side, plasma sample is placed and on the other, a non-protein drug-free buffer solution is placed. The unbound free fraction of the drug transverses across the semi-permeable membrane until at equilibrium the concentration of the free fraction of the drug across the two membranes become equal. The fraction of drug is readily determined by measuring the drug concentration on either side of the membrane. If the binding of drugs to the plasma protein is dependent on the drug concentration, then it could cause difficulties in assessment, as the drug concentration is lower on the plasma side at equilibrium as compared to the initial concentration due to diffusion of the free drug and also due to dilution of plasma by water, which transverses through the plasma side due to osmotic gradients. This is the main disadvantage of this method, but efforts are being made to overcome the osmotic effect by using previously diluted plasma. The second method is the ultracentrifugation method. This method does not alter the unbound drug concentration and hence does not disturb the equilibrium. It employs a centrifugation technique to separate the small unbound fraction of the drug from the high molecular weight drug protein complex. This method uses high gravitational force to separate the two. After centrifugation, the area just below the lipid layer of the very low density lipoprotein and chylomicrons is sampled and the concentration of free compound is determined, while the concentration of the total compound is assessed before centrifugation. Therefore, concentration of the bound drug can be assessed from these measurements. Contrary to the equilibrium dialysis, ultracentrifugation is a preferred method theoretically. This method also possesses some disadvantages, such as it is a time consuming method. If the drug binds to the low density lipoproteins, then during centrifugation these low density lipoproteins rise to the top of the tube, and thirdly, this technique is expensive as compared to equilibrium dialysis. The third method is the ultrafiltration method. It is another useful technique to measure plasma protein binding. It can be achieved by two processes, first being centrifugation and second by positive pressure. The filtration process employs a whisking tube for filtration of plasma water by placing it under positive pressure. Unlike the other two techniques, this technique is rapid and quite simple. This method has been used to assay the binding of antibiotics to the serum proteins. The major disadvantage of ultrafiltration is that some drugs bind extensively to ultrafilters, making it a cumbersome process. The next method is binding to albumin microspheres. In this method, initially albumin microspheres are developed and these microspheres are then transferred to a 5 ml plastic syringe containing buffer with a porous disc at the bottom, and drug candidate is added to these syringe containing buffer microsphere suspension. After administration of the drug in the suspension, these syringes are incubated at room temperature so that equilibrium can be maintained between the drug and the albumin. Now, this solution is pushed out of the syringes through the porous disc such that the unbound fraction of the drug is obtained and the bound fraction remains on top of the disc. This method is generally employed in cases where a drug candidate is suspected to bind to the albumin or to check any interactions between the drug at the binding sites of albumin. The advantage offered by this method is that it is a rapid and a simple method, and the disadvantage of this method is that it is only used to detect the albumin binding of the drugs, and uncontrolled temperature also remains an unresolved issue with this method. The next method is the gel filtration method. It is very efficient as it allows to measure the extent of plasma protein binding of the drug in a very small plasma volume. Gel filtration is based on the principle of simple chromatography and makes use of a chromatographic column, and the drug concentration is measured in the elute. The column is equilibrated with the drug so as to prevent dissociation of the drug protein complex. Initially, the binding protein is saturated with the drug and then it is passed through the previously equilibrated column with the same drug. The drug concentration is interpreted in the elute. The major disadvantage of this method is its complexity and time consumption. Table 2 summarizes the different types of techniques employed in the measurement of plasma protein binding as coming on the screen. Now, let us understand the kinetics of drug binding. It is a dynamic process. Kinetics of reversible drug protein binding can be described by applying the principle of law of mass action to the protein binding of the drugs. According to this, P plus D will give us a complex which is PD. P represents protein, D represents drug. So, we get a complex which is protein plus drug. When a drug binds to a protein, an association constant Ka is there, which is expressed as the ratio of the molar concentration of products and the molar concentration of the reactants. Thus, Ka is equal to Pd divided by P into D. Rearranging this equation will give us Pd is equal to Ka P into D, where P denotes the concentration of free protein, D denotes the concentration of free drug, Pd denotes the concentration of protein drug complex, Ka denotes the association constant, and Kd denotes the dissociation rate constant. The extent of drug protein complex formed is dependent on the association binding constant Ka. Drugs strongly bound to the plasma proteins have a large Kaa. In case of a forward reaction, Ka is greater than Kd, that is, protein binding is favored. In the in vitro experiments, pure albumin is used as a standard protein source, since this is majorly responsible for the binding of the drugs. To study the binding behavior of a drug, a determinable quantity R is defined, which is the number of moles of bound drug upon total moles of the proteins. The equation 5 and 6 are applicable to the situation when there is a sole protein binding site and the protein and the drug are in equal proportion in the protein drug complex. This assumes one independent binding site for each mole of the drug. In some instances, protein binding sites are more than one, or let us assume there are n number of binding sites available per mole of the protein, then the equation could be rewritten as the one shown as 7, where n is equal to the number of binding sites and R is the number of moles of drug bound upon total moles of protein. If more than one type of binding sites are available, then equation 8 is applicable, where numerical subscript represents different type of binding sites and K represents the binding constant. N represents the number of binding sites per mole of the protein. Determination of binding constants and binding sites by the graphical methods. The values of association constant and the number of binding sites can be obtained by various methods. The first are the in vitro methods. In the in vitro methods, both the values of association constant and the number of binding sites n can be obtained using the direct plot, the scattered plot, or the double reciprocal plot. Direct plot, it is a simple plot which can be made by plotting R versus D. The binding parameters can easily be calculated, indicating the number of binding sites on each protein. Scattered plot, this spread the data to give better line for estimation of binding constant and binding sites. By converting the equation 7 into a linear form, this plot is obtained as shown on the screen. The next is a double reciprocal plot. It is also called as the Line-Weaver-Burk plot, and this plot is made by the reciprocal of the equation 7. A plot of 1 by R versus 1 by D is made, which gives us a straight line with the slope 1 n by Ka and the y intercept 1 by n. Besides the various in vitro methods, in vivo methods are also used. The percent of drug bound is often used to describe the extent of drug protein binding in plasma. The fraction of the drug bound can be determined experimentally and is equal to the ratio of the concentration of the bound drug and the total concentration in plasma. Now, let us understand the clinical significance of protein drug binding. Number 1, it prolongs the residence time of the drug in the body. The drug bound to plasma proteins will not get filtered, as plasma proteins are not a normal component of the glomerular filtrate. Therefore, biological half-lives of the drugs which gets essentially eliminated by the glomerular filtration could be increased when subjected to protein binding. For example, peptides are considered as an important class of molecules for the development of therapeutics, as they possess certain properties like comparatively small mass, exceptional target specificity, high binding affinity and low toxicity. But their short half-life, which is only a few minutes, has hampered the development of a large number of therapeutics from peptides. Enzymatic degradation and fast renal clearance are the major reasons for the fast elimination of peptides from the circulation, which can be circumvented by fusion of peptides to the various plasma proteins like albumin or immunoglobulin, which results in the prolonged residence of the peptides in the body. Protein binding also reduces the rate of elimination. The unbound or the free fraction of the drug is readily eliminated from the body. Drugs which are extensively bound to the proteins are therefore eliminated slowly from the body, that is, their elimination half-lives are much longer as compared to the unbound drug. It is due to the fact that the protein bound drug is restricted to enter the liver because of its large molecular size. It is due to this large size only, it is also restricted to enter the glomerulus. For example, doxycycline is extensively protein bound and have an elimination half-life of 15.1 hours as compared to the 8.5 hours of tetracycline, which is 65% protein bound. There are examples of a large number of drugs whose elimination rates have been reduced due to excessive plasma protein binding. Drugs that are extensively bound to the plasma protein also results in high blood levels and low volume of distribution, which suggests that the concentration of drug in the blood as compared to that at the site of action will be high. For example, warfarin extensively binds to the plasma protein up to 99% and therefore has a low volume of distribution, whereas theophylline is moderately bound to the plasma protein and has a high volume of distribution. Another significance of this plasma protein binding is that we all know that the bacterial infections occur between the cells of an organ. Efficacy of a drug is due to its presence in a higher concentration at the site of infection. Therefore, an antibiotic which extensively binds to the plasma protein will be largely restricted to the blood and will be present in low concentration at the site of infection. Contrary to this, other antibiotic having similar effect but does not bind to the plasma protein is free to be distributed and may be present at the site of infection in a higher concentration. Such an antibiotic, despite of the fact that its blood concentration will be lower, is more effective than the first antibiotic in clinical situations. Competitive binding. Competition for binding sites over different proteins may occur such that the amount of bound drug may be decreased significantly by the presence of another drug present in the circulation. As it is known that the presence of protein binding largely affects the activity, distribution and elimination process, so competitive binding can result in an increased rate of biotransformation and elimination of the displaced drug. This displaced phenomena could be used successfully in case of intoxication to reduce the concentration of a toxic drug rapidly. There are many examples of these displacement reactions like warfarin and phenylbutazone. The latter readily displaces the former from its binding site, resulting in an increased concentration of the former. The next is dose alteration. Plasma protein may also be responsible for the rate and extent to which a drug reaches its site of action. For example, the two potent analgesics, morphine and oxycodone, both act through the same receptors, but as determined in the in vitro experiments, morphine show a 26-fold greater affinity for the opioid receptors than oxycodone. According to this finding, it could be assumed that the dose required for morphine should be less as compared to oxydone to exhibit the same pharmacological action. But the patient studies have indicated that following intravenous injection of both the drugs, equal doses were required to relieve the pain. This is because morphine readily binds to the plasma protein, therefore, there is a need to increase its dose. Plasma protein binding has also been used for diagnosis of various diseases in the recent past. For example, chloroquine is a drug which interacts readily with the melanin of the eye. The chlorine atom of this drug is replaced with a radio labeled iodine so that it can be used to visualize any melanomas of the eye. Now, let us summarize this module. An administered drug can interact with a variety of blood and extravascular tissue components, out of which drug binding to plasma protein is the most significant. Plasma protein binding can augment or diminish a drug performance in the body. There are a variety of plasma proteins in the body which binds to different types of drugs such as sulfonamides, anticoagulants, NSAIDs and several compounds used in cancer chemotherapy. The drugs bind to the plasma proteins in the following order. Albumin shows the most, followed by alpha acid glycoproteins, then lipoproteins and then globulins. The extent to which a drug is bound to a plasma protein is of great significance and can be measured by different techniques such as equilibrium dialysis, ultrafiltration, ultracentrifugation, gel filtration, etc. Plasma protein binding plays a significant role in absorption of various insoluble drugs, diagnosis of several diseases, dose alteration of various drugs, etc. Thank you.